Hi, my name is Emiliano Flores, and I work in the Bio5 Institute in Dr. Anita Koshi's lab, where we study Toxoplasma Gondii. Toxoplasma is one of the world's most successful parasites and infects up to 1/3 of the world's population, although it does not present any symptoms unless the host is immunosuppressed. There are two forms of the parasite, the bradyzoid and the tachyzoid. The tachyzoid can be available during acute infection and is a fast replicating form of the parasite. The bradyzoid is a slow replicating form, and it can be found in cysts where it can be found in neurons. Some of my beginning questions are, what mechanisms does toxo use to move through the brain? How can we track toxo through the brain? And what type of neurons does toxo in cysts? We know that the parasite moves with actin myosin motors, which gives it its gliding motility to move through the brain and into other cells. As of right now, we only know that toxo interacts with medium spiny neurons, but have yet to figure out the rest of the neurons. We can track toxo through immunofluorescence, which is the use of secondary and primary antibodies to see where the parasite has traveled within the brain. My project's main question is, does Toxoplasma Gondii co-localize with medium spiny neurons both in the striatum and in the cortex? 2 years ago, I started on this project by co-localizing parvalbumin with toxoplasma-infected neurons. We came to the conclusion that no parvalbumin neurons co-localized with toxoplasma-infected neurons, localizing toxoplasma-infected neurons with other subtypes of neurons in the brain. To do this, we use immunofluorescence as well as our transgenic mice, which we have created the Toxoplasma Cre system. The Toxoplasma Cre system is parasites with the Cre recombinase inject their Cre recombinase into the neuron, which excises a stop codon, which allows a promoter to express a GFP. Therefore, with the expression of GFP, the neuron fluoresces a bright green, which we can see under the microscope. Immunohistochemistry is the use of primary and secondary antibodies to be able to bind onto an antigen and see the fluorescence through a microscope. We image these pictures by using a confocal microscope, which allows us to dissect plane by plane with laser and light to be able to see our sections more clearly. And then they are quantified using a cell counter. Some of my ending questions are, does Toxoplasma Gondii interact with other subtypes of neurons? Does Toxoplasma Gondii interact with FOXP2 neurons? And can we potentially integrate other proteins into Toxoplasma Gondii so we can inject it into other neurons?